So now that we've talked about the cell membrane, we're going to talk about organelles. Organelle. Tiny organ. These are like the tiny organs of a cell. They're the functional units of a cell, and each individual part plays a role in cell function. So eukaryotic cells contain organelles with specialized functions. The nucleus, ER, and golgi interact to secrete substances such as milk proteins. These are some of your membrane-bound organelles because they're made up of membranes. So we'll start by talking about the nucleus. The nucleus stores DNA. That's its job. It's a storage container. A lot of people say the nucleus is the brain of the cell. That's not true. The nucleus doesn't think about stuff. Now there's processes that happen in the nucleus, but that's all regulated by enzymes. It's not like the nucleus is a brain and it's thinking about stuff. Things are going on in there, and all of those actions are mediated by enzymes. All the nucleus is basically a storage container for DNA. So the nucleus stores DNA. And there's a couple different parts to the nucleus. First, the nuclear envelope. The nuclear envelope is the boundary of the nucleus. The nuclear envelope is much more selectively permeable than the cell membrane. Why do you think that might be? Well, if anything gets into the nucleus, now you're messing with the genetic code, the directions for making everything that cell needs to survive. And maybe not only that cell, but making stuff for the entire multicellular organism, if it happens to be a multicellular organism. So we need to make sure that we really protect this DNA material. So we have nuclear pores, and nuclear pores are openings in the nuclear envelope that allow for substances to pass into and out of the nucleus. These are very careful in what they allow to pass into and out of the nucleus. Unfortunately, some viruses have mutated to now have the ability to get their genetic material right through your nuclear pores. So certain viruses can now cause cancer. Ribosome. So I just talked about a ribosome before. A ribosome is responsible for protein production. There's two different types of ribosomes, and it's not like when a ribosome is free or attached, it's going to be that for the rest of its little ribosome life. Free and attached, one ribosome can attach, one ribosome can become free, so it can switch between the two forms. But a free ribosome is floating in the cytosol of a cell. So here's where I'm going to take a second to define what cytoplasm is versus cytosol. Cytoplasm is talking about everything that's inside of the membrane. So that includes organelles and the cytosol. While cytosol is talking about the liquid portion only. So a free ribosome is floating around in the cytosol. An attached ribosome is attached to rough endoplasmic reticulum. And there's a difference between rough ER, rough endoplasmic reticulum, and smooth endoplasmic reticulum. Rough ER has ribosomes, and it's responsible for protein production to produce proteins that are going to be secreted from the cell. Smooth ER is responsible for making hormones, for lipid metabolism, and for detoxification of the cell. Within the endoplasmic reticulum, if there happens to be a region that has ribosomes attached, it's called rough. If there happens to be a region that doesn't have ribosomes attached, it's called smooth. Ribosomes can stick and fall off and stick and fall off, and if they're attached, the endoplasmic reticulum is going to be producing proteins, but if they fall off, the endoplasmic reticulum is going to become smooth, and then it's going to be responsible for lipid metabolism, hormone production, and so forth. So each section, it's not like this has to be rough forever. As long as the ribosomes are there, it'll be rough. If they fall off, it'll be smooth. And depending on the needs of the cell, this can change. So if a cell needs more protein, more ribosomes are going to be stuck to here, and that's going to be protein that's going to be secreted out of the cell or incorporated into the cell membrane. Now, if the cell needs more fats or wants to produce hormones, then these can fall off, and those would be certain types of hormones, but these will fall off, and then you'll have more smooth ER to perform that function. Vesicles are transport vehicles. So vesicles are membrane-bound organelles that transport substances around in a cell. And then we have the Golgi apparatus. The Golgi apparatus packages and modifies substances to be shipped out of the cell. So here's a picture of the Golgi. Substances go in from the endoplasmic reticulum, they get modified, activated sometimes, and then they go into a vesicle until they get secreted from the cell. The proteins leave the Golgi in vesicles, which fuse with the cell membrane, expelling their contents. So before we go on, I want to give you an analogy. So on this side, I'm going to have what actually happens when we produce a protein. And on this side of things, I'm going to have an analogy. So imagine you want to bake a cake for your aunt in Arizona. So you, as a person, want to bake this cake. You're going to go into your kitchen, and you're going to have to go to your storage cabinet where you have this giant recipe book. And this giant recipe book contains all of the recipes to make every single thing that you could possibly think of ever eating, ever. So in your kitchen cabinet, in your nucleus, you have a cookbook. Or you have DNA that contains individual recipes or genes. Now you want to find the recipe to make a cake. So you want to find the right gene to produce a protein. And I've come back to this analogy when we get to protein production, but basically you can't haul this giant cookbook onto the kitchen counter. All you can do is flip to the page where it has the recipe that contains the recipe for the cake that you want to make, and you're going to make a copy of it. So you're just going to jot down the recipe. You're going to take notes, and then you're going to take those notes to the kitchen to actually produce the protein or the cake. So now we've got our recipe figured out. Now you're going to make the cake. So the ribosome is you. And you're doing this in your kitchen, or the rough endoplasmic reticulum. Now what you're going to do is you're going to be reading that recipe and incorporating in the correct ingredients to make the cake. And the location that this happens to be going on is in the kitchen. So what's happening is the ribosome is reading the recipe that was incorporated in those genes that are found in your DNA, And as the ribosome is reading the directions, it's incorporating the correct amino acids in order to make a protein. Now, the location that this is being created is in the rough endoplasmic reticulum. So now your cake's made. So you're going to have to get in your car and drive to the post office. Because you want to ship it down to Arizona, you need to put it in a box and everything. You got to get it ready for shipment. Well, after your protein's made in the rough endoplasmic reticulum, it's going to be packaged in a vesicle, and then it's going to be taken to the Golgi apparatus. If you recall what I said the function of the Golgi apparatus is, I said it packages and modifies substances to be shipped out of the cell. It's kind of like a post office. So at the post office, you put it in a box, put an address label on, and so forth, and get it ready for shipment. So then you're going to go in the mail truck to your aunt in Arizona. Well, not you. The cake will be. But anyways, you get the point. So mail truck, and you get delivered to your aunt in Arizona. So from the Golgi apparatus, it goes into another vesicle, and then it'll eventually get shipped to the cell surface. So I am going to come back to this analogy again, like I said, with the protein production stuff that we'll talk about later on, but hopefully this gives you kind of an idea of what's going on here. So other types of organelles, we have lysosomes. Lysosomes, I like to call the mini stomachs of the cell. So lysosomes contain digestive enzymes, and they're going to help break down substances that are brought into the cell that need to be degraded. Sometimes you have dead organelles, and those dead organelles, you don't want those to be around and take up space inside of the cell. So you have to break them down, and lysosomes contain the digestive enzymes to do so. Also, if you have a white blood cell that just ate a bacterium in your body, in order to digest that bacterium, you need a mini stomach. You need a lysosome with those digestive enzymes. Peroxisomes contain antioxidants. When we go through the process of cellular respiration later on in the semester, you're going to learn about what happens when we produce energy. And one of the byproducts of energy production are oxidizing agents. They're called free oxygen radicals. Now these things are known as electron snatchers. They go around in your cell and they snatch electrons from things, which causes chaos inside of your cell. So what antioxidants do is they say, wait a minute, don't steal electrons. Here's a bunch of electrons that you can have to make you happy. So it's kind of like if a burglar comes in and wants to steal everybody's money, and then you got like this super rich person that stands up and says, hey, just don't hurt anybody, don't take anybody's money, here's a bunch of money, now go away peacefully. It's kind of what antioxidants are going to do. So peroxisomes contain these. You can never possibly consume enough antioxidants in your life. These really prevent aging, and it sounds, kind of like a cosmetic issue, but it's really not. They prevent aging of all of your organs, and they really help keep you healthy. So the more antioxidants you can have in the diet, the better. Mitochondria. Mitochondria are where a majority of the cell's energy is produced. Now, what they think about mitochondria is this endosymbiont theory. They believe that mitochondria used to be bacterial cells that lived outside, that were free-floating around in the environment. One day, this giant eukaryotic cell sucked up these bacterial cells. And the bacterial cells started doing work for that eukaryotic cell. And with that work, these eukaryotic cells thrived like crazy. So they're like, okay, you can stay. As long as you're doing that work, I'll give you protection. The bacteria were happy because they were now protected, and the eukaryotic cell was happy because it now got energy. Let's imagine that your best friend is a fugitive, and they're like running from the cops, and you hear sirens coming, and all of a sudden you hear a knock on your door. You open the door, it's your best friend, you're like, what's up? And they're like, hey, you gotta hide me, the cops are out to get me, blah, blah, blah. So you let them come into your house. And you're like, okay, I will give you protection, I'll hide you, but you're gonna do some work for me. You're not gonna live here for free. You're gonna do my laundry, I want meals cooked, I want you to take care of my kids, whatever. So that's kind of what happened with this endosymbiont theory. Endo means inside of, symbiont means that two organisms are living closely together. And this is a theory. And theories are based on a lot of evidence. So it's not a law yet, but it is a theory because there's a lot of evidence that supports this. So what are the pieces of evidence that support the endosymbiont theory? We didn't just create this because it sounded cool. And like scientists didn't have a lot of fugitive friends and were like, hey, this is a scenario that we could go with. There's evidence. So what's the evidence? First of all, mitochondria have their own DNA. Now, as a person, you can only get mitochondrial DNA from your mom. So if they do mitochondrial DNA typing on you, you are 100% match with your mother. Because if you think about what made you, your dad's sperm cell and your mom's egg cell, which, yeah, that's disgusting and all, but the sperm is just a little tiny cell that contains just half the genetic material, and it has a little tail on it. The egg contains all of the organelles. So the mitochondria in there comes from the egg, and that comes from your mom, and that can only come from your mom. So all of this mitochondrial DNA in here comes from your mom. But the mitochondrial DNA is circular. And guess what? Bacterial DNA is circular. The other thing is mitochondria have their own ribosomes, so they can produce their own proteins. That has a major role in human disease, and there's a lot of diseases that have been linked directly to mitochondrial dysfunction and the genes that are present in mitochondrial DNA, particularly like Parkinson's and Alzheimer's disease. But with these ribosomes, they're small. Like bacterial ribosomes have a different size than eukaryotic ribosomes. Bacterial ribosomes are smaller. So these ribosomes are bacterial ribosomes. The other piece of evidence is these guys can replicate all by themselves. So within your cell, they can be making copies of themselves independent of the cell cycle and cell replication. The last piece of evidence that I'm going to talk about is the fact that these have two membranes. The inner membrane is thought to have been the original bacterial cell membrane, while this outer membrane is thought to be the eukaryotic cell. When this eukaryotic cell was consuming or taking in these bacterial cells, they wrapped their own phospholipid bilayer around it as they brought it into the cell. And that commonly happens during cell eating, known as phagocytosis, which we'll learn about in the transport lecture next week. So that's the endosymbiont theory. Basically states that we think that some organelles used to be free living bacterial cells that all of a sudden got taken up by a larger eukaryotic cell, and then there was this symbiotic mutualistic relationship where they both benefited, so they kind of evolved to be together. This isn't just true for mitochondria, it's also the case for chloroplasts. So chloroplasts have those same pieces of evidence that lump them into this endosymbiont theory as well. So since I just mentioned chloroplasts, now we'll take a moment to talk about a few of the cell structures that are specific to plant cells. So plant cells, if you look at them, they have a lot of the same structures as animal cells. But plant cells are going to have large vacuoles. These are much larger than vesicles. And vacuoles, like plants typically have a central vacuole. They store nutrients, water, pigments, waste products. They are responsible for turgor pressure. Toxic substances can be used for protection from herbivores, so they can store a whole bunch of toxins. That's why some plants are poisonous, especially to cats. If you have household plants, you shouldn't let your cats be chewing on certain plants because it can kill them. Poinsettias are one of them. And then we have like some functions that are performed by lysosomes and other eukaryotes. The vacuoles actually contain some digestive enzymes in certain cases. So this central vacuole can be up to 90% of the volume of some cells. So plant cells are kind of known for their large storage vacuoles. And here's a picture of one of those here. You can see how big this storage vacuole is. And this is a real electron micrograph picture of a plant cell, and you can see how large this is. The next one is chloroplasts. Chloroplasts are responsible for photosynthesis. A lot of people think that photosynthesis is how plant cells get energy, and that's not true. Photo means light, synthesis means that you're making something. What photosynthesis is, it's the process of producing a sugar. So it's how plants make their own food, and it's how certain types of cyanobacteria and algae make their own food. So in photosynthesis, all they're doing is making food using sunlight as an energy source. Then they have to take that food and break it down in order to get energy. So they make their own food and then they break it down to get energy. And that energy originally comes from the sun, so that's why they end up with a net gain of energy production after that all of that happens. So a chloroplast forms discs that are called thylakoid discs. If we look inside here, all of these like stacks of pancakes, each one of those is called a thylakoid disc. They're stacked to form grana. So grana are the actual stacks that you see. So one stack of pancakes is called a grana. Each individual component of that stack is called a thylakoid disc. And then they're suspended in a fluid known as stroma. So stroma is the fluid that fills up the inside of a chloroplast. So those are some of the unique structures that you'd find in plant cells.